Okay, we have the great pleasure of having a special guest with us today. James Bircher is a present to us by Evogene, who has him here for research and consulting with them, and so we were able to kidnap him and bring him here to us to talk to us about some of the research that he's doing. And since we're way behind schedule, I think Jim's talk will speak for itself. He's one of the great biologists, the great geneticists in plant sciences, and he will tell us some actual basic science here. Thank you. Jay.
Well, thanks very much for the invitation to speak here today. I suspect that my talk's going to be very different from many of the others. It's going to be a scientific talk. I've tried to modify it for the audience to some degree. Certainly food security is something that is the driving force behind what I'm going to tell you about from a societal point of view. And the particular thing that I'm going to talk about is to an attempt on the beginnings of the development of a way to take the genetic material and basically reorganize it the way that we want to, sort of a synthetic biology approach or artificial chromosome types. Of course, the chromosomes are the genetic material, and this is the basis of how we've developed resistances to many diseases that have helped in food security. And what we want to do is to, as one of the speakers this morning was talking about, looking ahead in terms of decades, and perhaps what I'm going to tell you about is a projection that will take a few decades before it will have serious applications, but that is what our hope is. So not only are we interested in developing a technique that will be potentially useful in food crops, but I just want to take a few minutes to talk about some other issues that I think are a need to be a complement to new technologies in agriculture and biotechnology. One of them is there is a need to preserve the genetic diversity of the crops that we do have and the related species that can donate genetic material. This has been useful in agriculture over many decades, and we need to think ahead in preserving that diversity of the genetic material. Another issue that has not received a lot of attention, but I think is one that deserves some attention, and that is that there is perhaps a need to diversify the food crops that we have. The current ones come from a very narrow base. In human history, hundreds of plants have been used for food, but currently, as you've seen in the other talks, there's only about a dozen that constitute the vast contribution of calories. And so in terms of developing the biotechnology that I'm going to tell you about, another thing that is a complement to it, which remarkably, as I've done some research on this recently, there's very little known about it, and that is that in terms of the metabolites and proteins that may be useful in a biotechnological point of view, that may have antifungal, bacterial, or insecticidal properties, we know very little about these metabolites and proteins. We know some of them, and some of them have been used, of course, but many others of them we know very little about. So then what we'll talk about is diversifying crop properties using genetic engineering and synthetic biology. And of course, the complement to that is that we need more efficient genetic engineering techniques in general. So this is the public's view of genetic engineering. You just inject something into the crop and you miraculously have something that's beneficial. In reality, it's much more complicated than that. And the specific thing that I'll tell you about is our initial attempts to make artificial or so-called synthetic chromosomes. So why would one want to do this? So some of the issues is that it will give a more controlled expression of genes that are put into an organism. Of course, already there are herbicide resistance and insect resistances, and these perhaps could be improved and expanded. There's currently a big push for doing biotechnology and drought tolerance and nitrogen utilization. There's also efforts to improve the photosynthetic efficiency of rice and to use various plants as biofuels and biodiesel in canola and soy. And, of course, there are efforts to improve the nutritional composition, particularly in cassava in the third world. So the potential of developing synthetic chromosomes would be that one might have in the future the ability to add whole biochemical pathways to plants for new properties. You could do combinations of all of the above, potentially the mass production of useful proteins, and from a medicinal point of view, it may lead to the mass production of plant pharmaceuticals. So in developing this kind of mini-chromosomes, the small units of heredity, as we mentioned, one of the problems in putting new genes into plants is the variability of expression. And so if we had these chromosomes that were basically synthetic, we could use that as a platform for the faithful expression of many genes that could be placed into a plant. As also we mentioned, they could be used potentially for whole biochemical pathways to add new properties to plants looking into the future. So what this could do then is that if we have it on an independent chromosome, it would allow plants to be used as a factory for the potential of multiple foreign proteins or other useful metabolites. So artificial chromosomes have been produced in mammalian species, and there have been two approaches that have been taken. One of them is to take the part of the chromosome that is involved in the chromosome's inheritance, which is called the centromere, and take those sequences, put them back into a cell, and they aggregate into very large pieces that sometimes will function. And we'll come back to that point in terms of how it does or does not work in plants in just a minute. The other way is to take the very ends of chromosomes, which are specialized DNA sequences that stabilize the ends of chromosomes. These are called telomeres, and if you introduce them into a plant, then this will actually cleave off the chromosome. And as you'll see, one can cleave off basically all parts of the chromosome, and at the same time you can put in whatever genes that you want to do at the same time. So in terms of using centromeres as the part that is involved in the inheritance of chromosomes in plants, this has been tried in the academic world, and in every case where it has been tried, you can take those sequences, put them into a plant, and they do not function as a centromere. And so the sequence of the DNA actually is not responsible for its function, and we say, therefore, that it has an epigenetic as opposed to a genetic property for its functioning. And so there's a large literature to show that the centromeres that can become inactive, or they can not act when they are put back into a plant. So this approach has not been one that has been very useful. So the obstacles that we were interested in trying to overcome were this epigenetic component of centromere specification, and at the time we started this, there was no efficient homologous gene targeting system, and the efficiency of gene homologous targeting, in other words, to put a gene where one might want to in a plant, still can be improved in its efficiency. So what we did was we took the approach of putting telomeres back into a plant to cleave off basically everything on the chromosome using the endogenous centromeres, which are still in the cell and still functioning as they might. So we use the telomere-mediated truncation to produce these. So when we transform a plant, put those sequences into the cell, the presence of a telomere basically will cleave off the chromosome arm, and we can do this with the common way of transforming plants is with agrobacterium transformation. That carried multiple copies of the DNA sequence that encodes the telomeres. And the truncations of the chromosomes can be recognized by a cytological assay without having any intervening haploid stage, which would select against the large deficiencies that would be produced. So this is the way that we did it. We basically produced constructs that would have all of the necessary features to recognize transformations and then place telomere repeats at one end. And these were transformed into the plants. At a certain frequency, they would cleave the chromosomes. And this is the approach that, in terms of a diagram, how it works. So we have a construct, has a telomere repeat in it. This is a Maize chromosome. Here's the centromeres. Here's the telomeres, the ends of chromosomes. When one puts a piece of DNA in, there's a double-strand break in the DNA, and if that piece that we are adding inserts in there, it can be a stable insertion, or if there is truncation, then that piece is lost, and at the very end of the chromosome, what we've added to the cell is now placed at that position. And so if we are sufficiently efficient in our transformations, we can basically cleave off most of the chromosome arms. So here's some examples of the chromosomes. You can see here, this is the little red dots here, if you can see that well enough under this lighting. That's the genes that we added to the chromosome, and the rest of the chromosome arm has been eliminated from it. And so one can do this fairly readily. Here's another example here in which the whole chromosome arm has been deleted. The little red spots here are the genes that we've added to it. This is the centromere of the chromosome, so we basically eliminated the whole chromosome arm. And this was one of the first ones that we analyzed, and we could put it then through a series of crosses into, turned out it was in a diploid, or in a tetraploid. We crossed it into a diploid, and then we could do genetic manipulations with it, which I won't go into detail. So one of the other things that we wanted to do was to use a separate type of chromosome in maize called the B chromosome. These chromosomes basically are dead chromosomes. They are in a few thousand species of plants. No one really knows how they're formed. But they turn out to be very useful for this purpose because they have no known genes that might be carried along. And so we wanted to see if we could take the B chromosome and make mini-chromosomes from it by transforming in the telomeres. And so in this case, we bombarded in the telomere constructs into recipient cells that, of course, had the normal chromosomes, the so-called A chromosomes, plus the supernumerary B chromosomes that could be cleaved off very easily with no consequences. And they have this additional advantage that the B chromosomes have an accumulation mechanism that can perpetuate them despite the fact that they have no genetic material in them, and we can use that, as you'll see, to manipulate the dosage of them. So we can do a bialistic transformation. We have an expression marker, and we have a target here of our chromosomes, and we can fire this in with bialistic transformation. And we can get then numerous truncations of the B chromosomes, which you can see here in this cell. There's actually two different truncations. This is a specific sequence on the B chromosome that we can distinguish them from all others. So there's different locations where the truncation has occurred, and you can see that with the red dots here. And this is the winner of the smallest one that we found, which basically has the centromere of the B chromosome present and the genes that we have added to them. And so we basically now have the endogenous centromere and our added genes to it on a separate chromosome. and the potential then to grow this chromosome back in the way that we decide to do so that's okay? So the gene that we had put onto the mini-chromosome to test for expression is expressed quite well relative to the control, and so our goal, as we have mentioned, is to develop a system where we can put genes into plants and have them expressed faithfully, and that on our initial study seems to be the case. So from those initial results, we were encouraged that we could do this further, and so we tried even other procedures of basically taking the genes that we were interested in and the sequences of the telomere, and we can basically mix them together and do a transformation. And when one does this, one can find mini-chromosomes that are derived from the A chromosomes that have cleaved off both arms and have basically nothing there but the centromere. We actually, for this particular chromosome, we've sequenced the whole genome again with a reference, and so we know that there's basically no genes there. It's simply the DNA sequences and a few flanking unique sites that are present there, plus the genes that we've added to it. So we have then achieved also with the A chromosomes, the normal chromosomes, the ability to make a tiny chromosome that has the potential to grow in future manipulations. And this slide just shows that that particular mini-chromosome has the normal endogenous DNA sequences present on it. So it's just basically a normal chromosome, as you see here, with all of the genetic information otherwise that has been removed from it. So the next question is, now that we have shown that we can produce these, what do we do to move forward? And one of the things that we want to move forward with it is to do modifications of them. And so that's one of the things that we have recently done. What we want to do is, this is perhaps for some of you a foreign language, but this is the selection marker that we use, and these are the telomeres that we use to cleave off the chromosome arm, and this is the centromere. So what we wanted to do is get rid of this gene so that we could use it again, and we have a promoter here, and so we have sequences here that in the presence of a particular enzyme will basically flip out what is present between those sites. It's called the Cre-Lox recombination, site-specific recombination system. And so the idea is that if we now have our mini-chromosome and we expose this chromosome to this recombinase, can we show that we can get rid of particular DNA sequences and leave this behind? And this is just another diagram of what would happen if you have the mini-chromosome with our particular sequences and you expose it to the Cre recombinase. Can you flip this out and be left with this behind? And indeed, this is what happens. So we have the Cre recombinase transformed into several lines of Maize, and we have our mini-chromosome here. And we can tell, actually, cytologically that this has been changed in terms of the intensity of the transgenes. And by looking at the molecular features of the transgenes, if one looks at the DNA properties of the transgene and looks at the sequences going across that selectable marker, and then if we add in the Cre recombinase, then we reduce the size of that in terms of the sequences that are present there. And in fact, we've sequenced this whole band here, and we know, in fact, that the reaction is behaving as we would anticipate that it might. So this shows then that we not only can make the mini-chromosomes and add new genes to them, but we have the ability to do an in vivo modification of them. And so we can do the co-bombardment. This can produce the engineered mini-chromosomes. The structure is complex. If we cross it to Cre recombinase, we can modify them in vivo. We can get rid of the selectable marker using these site-specific recombination systems. And this then serves as a proof of concept for in vivo modification. So in terms of what else can we do with these, one can do a dosage manipulation, and we've looked at the pairing and transmission considerations. With the mini B chromosomes, because it has this system for accumulation, we can add back normal B chromosomes to the line that has these mini B chromosomes, and each of these little green spots here is a mini chromosome that's carrying genetic information. So we have been able to run up the copy number of the mini chromosome to 19 copies in the presence of the normal B, which supplies the ability to increase the number of copies of the B chromosome. So the multiple mini-Bs, in terms of their pairing, they don't pair with each other. In this case, all of these mini-Bs are the little red copies here, which is an issue that needs to be considered in using them in biotechnology. But by using a B chromosome here, it's probably not possible to see it in this light. Using a larger truncated one, those show normal pairing and disjunction in meiosis. And so by using a longer B chromosome, one could immediately begin to use these in biotechnological applications. So simply some genetic issues. The small chromosomes usually cannot find their homologue in meiosis, and this then prevents 100% transmission. This is due to some issues in terms of the way that chromosomes behave, and those would have to be overcome for some types of biological applications currently without modifications. And so those are some of the issues that need to be addressed in this field. For these agronomic applications, those would have to be overcome. We are currently working on some ways to overcome them. One might be to put a restorer of fertility for cytoplasmic male sterility onto the mini chromosome. The prediction would be that then if we use a single one as a male parent, then 100% of the progeny would carry the mini chromosome and all of the genes that we've added to it. Similar systems occur in rice, and the potential exists that the similar situation may be able to be developed with wheat as well. So we think that in the future we can modify the mini chromosomes in a way that would make them agronomically feasible. And of course, as I've already mentioned, the alternative is to use a larger truncated engineered B chromosome, which basically could be used at the present. So no such modifications would be needed for breeding programs that are followed by vegetative propagation. Many of the important foods of the world have breeding programs that then are followed by vegetative propagation. Note that the mini chromosomes could be used as they are currently in this current state of how they work because they show excellent stability through development. And so this would potentially allow artificial chromosomes to facilitate the transfer of transgenes across diverse cytoplasms in breeding programs, and then they could be vegetatively propagated with their new properties. So the next step that we are currently addressing is to develop ways, and we are working on them currently, of adding even further genes to an engineered mini chromosome. And the potential exists then as you have gene 1, this is the centromere, the telomere, to keep adding genes to ad infinitum, to add and subtract them to how one might want to modify a particular crop. So in summary, Plant artificial chromosome platforms can be initiated by telomere-mediated truncation. The sequence of the telomeres is very similar, or in fact identical in almost all plants, and we know now from work of many others that it will work in many other species. Bypasses the epigenetic complications of centromeres, serves as a platform for predictable gene expression. We can add genes by site-specific recombination. I didn't talk about that, but it's been demonstrated. B chromosomes can be an excellent target. Highly deleted A chromosomes can be recovered. In vivo release of the selectable marker has been demonstrated. The potential for stacking a large number of transgenes is present there. We're also currently working to combine this with haploid breeding to transfer it to new lines very easily. It could be used for the mass production of pharmaceuticals or useful metabolites, add multiple new properties to plants, basic studies of chromosomes made to order. The telomere truncation system has now been shown to work in Arabidopsis, brassica, wheat, barley, and rice. So the 3 major crops of the world are on their way to having this technology. So as I like to say, that's the news from the Jim Maize Cafe. These are the people who have contributed to this work. There's a question over here.
Have you done any analysis of the whole plant level, the transcriptome or whatever, to see if these extra chromosomes have any other unintended effect on the plant metabolism, plant expression? I can imagine that it could be sequestering many other proteins that wouldn't have been different.
No, we have not done any of those transcriptome of the whole plant. Certainly on the phenotypic level, they don't seem to have any impact. If you run up the copy number of the mini-chromosomes to 19 copies, then the fertility begins to suffer a little bit. Presumably because there's 20 normal chromosomes in maize, and now you add another extra 19 chromosomes. So you basically, without adding genetic material that's pulling its weight and contributing to the constituents of the cell, you have, basically the same number of centromeres that are there that are perhaps affecting the way that the mitotic spindle is operating. It's an interesting basic question for which we don't know the answer. But on the 1 copy or 2 copy level, there's nothing that we can observe that just seems to have any impact upon them. Dr. Delmer.
Do you have any current collaboration on the vegetative trophy, like cassava or banana?
So we have not. We have had discussions with the Danforth Center about working on cassava, and we've developed a karyotype for cassava so that the potential is there to do it. We have not proceeded any further toward developing mini-chromosomes in cassava, although I would like to. They have their hands full with many other things, as you probably know. But I think that would be a very useful and important crop to work on, because the breeding of cassava is very difficult, as I understand, but then once one has a mini-chromosome with everything on it is an independent chromosome, then you can vegetatively propagate it and it has no problem whatsoever. So cassava would be a great system that would have utility right away. Exactly, and these are very important crops that have difficult breeding programs but are vegetatively propagated, and we could go straight into usefulness of this system in those species.
Thank you very much. You see hands all over the place, but we have to keep the schedule.
So
We'll have to get you